Hello everyone, myself Deepika Sharma, Assistant Professor Botany from the Guru Nanak Girls College, Yamunanagar. In this video, I am going to discuss about the life cycle of Penicillium, Part 1. This topic, it comes under the class BSc first semester. Life cycle of Penicillium, which is discussed in this video, it is mainly consist of the vegetative plant body of the Penicillium up to the asexual reproduction of the Penicillium. Now let us start with the main points that I am going to cover in the part 1. First we will discuss about the brief history of the Penicillium fungus, that is the wonder drug penicillin. The classification of the Penicillium, its systematic position, occurrence and habit of this fungus, and some of the common species of Penicillium which are found in our day-to-day life also, vegetative structure, that is the mycelium growth of the fungus, and reproduction, that is the asexual reproduction. Now let us start with the brief history of the Penicillium fungus. From the childhood days onwards, we have listened the name of the drug, the antibiotic penicillin injections. It's the first modern antibiotic drug. What do you mean by antibiotic? Antibiotic are those organic compounds which are synthesized from microorganisms and are used to retard the growth of certain disease causing microorganisms. This antibiotic drug, that is penicillin, was discovered by Alexander Fleming in 1928 from the species Penicillium notatum. One more species along with Penicillium notatum was also found more productive for the synthesis of this drug, that is the penicillin, from the fungus Penicillium chrysogenum. This antibiotic, it is used to prevent bacterial infections, means it is very effective for the diseases which are caused by bacteria like pneumococcus, staphylococcus, streptococcus, etc. Because in the presence of this antibiotic, bacteria fails to develop cell wall and are finally killed. And how do they prevent bacterial infection? Because they inhibit the enzyme which are responsible for the cell wall formation of Penicillium fungus. There are so many modified penicillins drug which are synthesized from the culture of this Penicillium fungus. One is, I have given the example penicillin Z, that is the benzyl penicillin. It is used as a intramuscular injection form, and some forms of the penicillin drug they are also used orally also. So this is all about the discovery of the penicillin, that is the wonder drug penicillin. Now the classification. The system of classification which is I have followed in this video is based on the classification system proposed by Ainsworth in 1972-73. Ainsworth divided the fungi into a kingdom, fungal kingdom, that is. What do you mean by fungi? It's a group of those plants which have a thalloid plant body, but they do not have any pigments, so they cannot prepare their own food, and their eukaryotic cell, well developed nucleus. These are the fungus characters. Division, Eumycota. What are the characteristic features of Eumycota? Eu means true. The true fungus in which the cell wall typically made up of chitinized, and the mycelium mainly multicellular but sometimes unicellular also, and eukaryotic, true nucleus is present. Subdivision, Ascomycotina. The name of this subdivision, it is based on the type of ascospores. They are perfect sexual stage spores which are found in the body asci. Now, along with these ascospores, some asexual spores or non-motile spores are also formed in this group, but they are not produced inside any body, that is sporangia. And third character of Ascomytuna group is aseptate, that the thallus is a septate mycelium. Class Plectomycetes. What are the characteristic features of the plectomycetes? The fruiting bodies, they are formed, which are called as a clistothecia. These specialized fruiting bodies, they are closed, in which asci they are present, which break down at maturity and release the ascospores. Two genera which belong to this group are Aspergillus and Penicillium. Order Aspergillales and the family Aspergillaceae. The name of this order and family, it is based on one of the genera which belongs to this group, Aspergillus. But now, because the perfect stage of the Aspergillus is discovered as a Eurotium, so now in some classification system, the order is also written as Eurotiales and family Eurotiaceae. Genus Penicillium. So this is the classification proposed by Ainsworth for Penicillium fungus. Now, what about the occurrence and habit of this Penicillium fungus? It's a large genus which comprise about more than 136 species, which are commonly called as green and blue mold. Most of the fungus, they are saprophytic in nature. What do you mean by saprophytic? Means the rotten, the fungus which depends upon the rotten, dead, decaying fruits, vegetables or any plant and animal remains. But some species are parasitic also, means they cause diseases. Why it is called as a green and blue mold? Because it forms a bluish green color mycelium on any substratum on which this fungus occurs. Now, Penicillium means brush. It is due to the presence of the chain of conidias which are formed on the tip of these branch conidiophores. These conidia, that non-motile spores, they are formed only during asexual reproduction. So here its resemblance is with the brush. That's why it is also called the broom fungus or a brush fungus. Next, there are some common species of Penicillium also. The two species that we have already discussed, they have antibiotic properties, and those are Penicillium notatum and Penicillium chrysogenum. They used to produce the drug, antibiotic drug, that is the penicillin. We've already discussed. Then the two more species of Penicillium, Penicillium roqueforti and Penicillium camemberti. They hydrolyze casein of butter and fats, and they also impart a tasty flavor and texture to the cheese. And here I have shown you the picture of Roquebrune cheese, which is mainly used which type of fungus? That is the Penicillium roqueforti. One more fungus of Penicillium, Penicillium purpureginum, it caused some spots on the papers and books. And here these two species, Penicillium italicum and Penicillium digitatum, they are responsible for the spoilage of citrus fruits. Penicillium italicum, it causes blue mold of the citrus fruit because it forms a bluish color mycelium growth on the citrus fruit. Penicillium digitatum, it is also called as a green mold of the citrus fruit because it forms a greenish color mycelium fur on the citrus fruits. And one more species is there, Penicillium expansum, which cause the rotting of apples, pears and grapefruits in the storage. So these are some of the common species of Penicillium. Now let us discuss the vegetative structure of Penicillium. The main plant body of the Penicillium, it consists of well developed multicellular mycelium. This mycelium, it can grow superficially, that is on the substratum surface, maybe citrus fruit, paper or anything on which this fungus grow, or sometimes it also penetrates deep into the substratum. Here it forms the nutrition absorbing organs like hostelria, which are useful for the absorption of nutrition. Now this mycelium, it's a brightly colored bluish green depending upon the type of species of Penicillium, hyaline or the pale, and it consists of a long thread-like tube-like structure which are highly branched, and these are called as hyphae. But the main characteristic of these hyphae is that they are septate. Now what do you mean by septa? Septa, these are the cross walls which divide the cytoplasm of a cell. Now here in this diagram, this is the septa with a simple pore, and this septa containing hyphal cell, that is the septate mycelium. Here this pore in this septa, it have a purpose of maintaining the cytoplasmic continuity from one cell to another. What does it mean? The cytoplasm from one cell can easily migrate to the another cell through the simple pores. Next, each cell, it is thin walled, short, and in earlier condition it is uninucleate, then it become binucleate, and later on a multinucleate condition will be there. Again, this single cell structure, the outermost layer is called cell wall, which is made up of fungus cellulose chitin. Just below the cell wall, there is one more layer, that is the plasma membrane. Inside the plasma membrane, a viscous fluid-like granular liquid is present, which is called as cytoplasm, in which the cell inclusion bodies are present. And the very prominent is nucleus. Nucleus, the true nucleus is present, due to which it is called as a eukaryotic cell. Ribosomes are present, which are called as a protein factories because they synthesize proteins. Next is the endoplasmic reticulum, ER, which are useful for the transportation of proteins, vacuole for the storage of cell sap, and the mitochondria, that is the powerhouse of the cell. Now, you know that fungus do not have any pigments, so they depend upon others for the absorption of their nutrition. That is, they have a heterotrophic mode of nutrition. In that case, whatever the food they absorb, it is stored as a reserve food material, and this reserve food material is oil globule. This is the diagrammatic representation of Penicillium, which show a hyphae. What are hyphaes? These are the single unit. There is a septate, tubular, branched units of mycelium, and some of the hyphae at the time of asexual reproduction, they grow vertically upright and get changed into the conidiophores, which bear a conidia. But these specialized structure, they bear born only during asexual reproduction. So this is the mycelium, which show a hyphae and the conidiophores. Next, the reproduction. Two types of reproductions are present in a Penicillium fungus. One is a asexual reproduction and another one is a sexual reproduction. What is asexual reproduction? In which no fusion of gametes take place, but mainly it is occur by the production of spores in the fungus. The one parent is involved, and in this fungus, only non-motile asexual spores are produced. What is sexual reproduction? Sexual reproduction is that mode of reproduction in which there is a fusion of gametes which are produced in their respective sex organs, that is, the male sex organ or the female sex organ. So this is the basic difference between the asexual reproduction and the sexual reproduction. Let us take the asexual reproduction first in detail. In this fungus, asexual reproduction, it takes place by 4 modes. Most of the species of Penicillium, they reproduce by the conidia formation. The second is fragmentation. Not only Penicillium, but fragmentation is also common in other group of fungus also. A very common mode of reproduction in fungus. Third, sclerotia. And the fourth one is the oidea formation. Let us take these asexual reproduction mode one by one in detail. Asexual reproduction by conidia formation. What are conidias? These are some non-motile, uninucleate spores which are produced in chain at the tip of special hyphal branches. They are bluish green in color. And why they are bluish green in color? Because they form a bluish green color of mycelium on the substratum, and it is also depend upon the species. Shape is globose to ovoid in shape. Color of the conidia is due to their walls. Each conidium has 2 wall layers. Outer conedial wall is thick, smooth or variously sculptured, and the inner wall is thin. These conidia, they borne on some specialized erect vertically upright hyphal branches which grow upright or vertically, and these specialized branches, they are called as the conidiophores. Now, how many types of conidiophores are there? Let us take the types of conidiophores. 2 types are there, monoverticillate and biverticillate. What is the basic difference between these two? Mono means single conidiophore without any branch. Unbranched conidiophores, they are called as monoverticillate. But these unbranched conidiophore, they bear a cluster of stigmata. What do you mean by stigmata? A flash-shaped bodies. And at the tip of stigmata, a chain of conidiospores are present. So these unbranched conidiophores with a cluster of stigmata and the chain of conidia on each stigmata, it is a monoverticillate type. What do you mean by the biverticillate type? Biverticillate type is a branch structure. The conidiophores have a branches, and each branch, it is divided into a medullae. Ultimate branches, these are called the medullae. Each medulla, see in the diagram, it bears a flash-shaped bodies, and these flash-shaped bodies, they then bear a chain of conidias. So here the conidiophores are branched. These conidiophore with the branch structure, then medullae, which are the ultimate branches, and the flash-shaped body stigmata, they are called as a biverticillate. In some species of Penicillium, monoverticillate type is present, and in most of the species of Penicillium, biverticillate type of conidiophores are present. So, the basic structure of conidiophore, it consists of 4 parts main. One is the conidiophore, which is actually a vegetative cell, but during the asexual reproduction, it grows vertically upright into a tubular long slender hypha, which is specialized for the formation of conidias. Second, medullae. The ultimate branches of the conidiophores, they are called as medullae. Each medulla bears a long flash or a bottle-shaped structure, which is uninucleate bodies with a tubular tip present on the medullae, and these flash-shaped or bottle-shaped bodies are called as a stigmata, Another name is also there, phyalides. These stigmata, each stigmata, they bear a long chain of conidia spores in some specialized order, that is the basipetal succession. What do you mean by basipetal succession? That older spores, they are present at the tip and the youngest at the base. So these are the main parts of the conidiophores. Now how do the formation of these conidia take place? These conidia, they form at the tip of stigmata, and you know that what is stigmata? It was a flash-shaped body which is present on the medullae if it is biverticillate, or directly on the conidiophore if it is a monoverticillate conidiophore. I already told you, stigmata, the flash body, they are uninucleate. The nucleus of stigmata divide into two nucleus, in which one migrates into the swollen tip of the stigmata and one is remaining in the stigmata. Then the terminal swelling secrete a conidial wall inside the wall of a tube and become converted into a conidia. In the same manner, next conidium is formed. Now the diagrammatic representation, that is the stages in the development of conidia, are shown in this pic that I have taken from your book. This is a vertically upright grown hypha, septate hypha, which is now called as a conidiophore because it is going to bear a conidias. In monoverticillate, directly these flash-shaped bodies, they are present. Whatever be the case, whether it is a monoverticillate or biverticillate, the formation of conidia is the same. These flash-shaped bodies, the stigmata, they are uninucleate. So here the nucleus is divided into two. One is migrated into this swollen tubular tip, and after that, this swollen tubular tip, it secretes a wall inside this tip, and the first conidium get formed. This conidium get pinched off from this tip, and after this, a next conidium start its formation just below the first one. And in this manner, a chain of conidia that could form on the tip of stigmata. But note on the main important point, the youngest is at the tip of the stigmata and oldest are now at the apex. And this order of the arrangement of conidias on the stigmata, this is called as a basipetal succession. Now, these conidias, they are very large in number, means each chain consists of more than 100 conidias which remain adherent with each other by some connective. And due to the presence of these conidia, this fungus appears like a broom or a brush. I already told you. And technically it is known as pencillus. In Latin language, pencillus means small brush. So here, the so many conidias, they are formed. After their formation, these conidias, they are disseminated by wind and they germinate on a suitable substratum if they got a favorable conditions, that is the favorable warm temperature and moist conditions. Each conidia have a two walls, I already told you, the outer and the inner wall, but they germinate by this septa formation, and this the outgrowth is there, formation of outgrowth will be there, and this outgrowth is called the germ tube. So these conidia, they give rise to one or more germ tubes. These germ tubes, they grow, become septate and transformed into a mature mycelium. So here you see there's the conidia which are produced in such a large number. They serve for the quick propagation of the species during the growing season unless it gets its favorable temperature or a moist condition. So these are the stages which are the development, they are showing the development of conidia, and this is the germinating conidia which formed a mature mycelium. So this is the main asexual reproduction, that is by the formation of conidia. Next, the type of asexual mode is the fragmentation. What do you mean by fragmentation? That mycelia, it get broken down into smaller pieces, small fragments. But each fragment, it is capable of independent existence. Means it can divide and it can grow and give rise to a complete new hypha. And this new hypha, which grow and transformed and get mature into a mycelium. This is the most common mode of reproduction in most of the fungus. That is the fragmentation. The third one, sclerotia formation. Now here the diagram of sclerosium. How does it look like? It's a compact mass of an interwoven hyphae. Means these hyphae of the mycelium, they are so tightly packed that they form an outer protective covering, which is also called as a rind. But this structure, it is dormant during unfavorable period. Means it does not grow during that period because it is not getting any favorable temperature or moist condition. So hence, sclerotia, they can act as a method of perennation. But once they get their favorable temperature and moisture condition, they germinate and grow into a mature mycelium. So this is the sclerotia formation. In Penicillium, mainly the sclerosium form, but there are other arrangements of fungal tissues are also there. But these fungal tissue arrangements, they are taken place in another fungus like prosenchyma, pseudoparenchyma and the rhizomorph-like structure. All these are the arrangements of the mycelium. Now the next, asexual reproduction by oidea formation, which is responsible for the torula condition in Penicillium. What are oidea? These are thin-walled spore-like structures. Now here this is the mycelium hyphae. And how do these oidea-like structure they are formed? They are formed from the hyphae by the formation of additional septa. Due to this septa, these non-motile spores, they are pinched off from this hypha. And later on they increase in their number for the formation of this bud-like structure, that is the budding. This structure, just remember as the structure of yeast, which also reproduce by budding. So here these oidea formation, which they start budding, they resemble a torula, and that's why it is called as a torula stage of fungus. But this oidea formation and the budding, it occurs in a sugary solution. So there are certain conditions in which this bud-like structures in the oidea are formed. Now the next, the conidia, not only the mycelium, even the conidia, if they are also taken, grow in a sugary solution, they fails to form then mycelium. But they germinate just like yeast, that is by budding. They also start budding like yeast, and in that case they produce a torula stage. So this torula stage condition of Penicillium fungus, it only occurs in a highly saccharine solution. But there is one important economic importance of this torula stage, that like yeast it also induces alcoholic fermentation. So oidea formation, which is not only act as a mode of asexual reproduction, but by budding they also induce alcoholic fermentation. So these are the 4 modes of asexual reproduction, but the most common among these is the conidia formation. The second one is a fragmentation, third one is a sclerotia, and the fourth one is a oidea. So this completes the asexual reproduction in a Penicillium fungus.